I'm here to talk about passive membrane transport. There's a couple different types of membrane transport in the two broadest senses, passive and active. Passive requires no energy, and that's what we'll talk about today. It can be divided into a few different subcategories: simple diffusion, facilitated diffusion, and osmosis. First, let me put those aside and direct us to what we're talking about with a cell. So in a cell, the membrane is this yellow line around the outside. And a very important feature about that membrane is that it is selectively permeable. It is a barrier to some things and not a barrier to others. So it's selective. Certain things it allows in and out freely, others not so much. So let's first take the case of simple diffusion. In the case of simple diffusion, the membrane is not a barrier. What sort of things can move freely in and out? To make a short list, there's oxygen, there's carbon dioxide, there are small molecules like ethanol that could move in and out, and lipids. Since the membrane is made of lipids, then it makes sense that lipids could move freely in and out. In diffusion, simply means that they move down their concentration gradient. Essentially, they spread out. So that's simple diffusion. Putting that aside for a moment. The more complicated one is facilitated diffusion. Now, facilitated diffusion is kind of everything else that a cell would typically deal with. This would be things like ions, anything with a charge, something like sodium ions. I'll illustrate that using a red bead. Glucose or other larger molecules, especially if they're polar, can't get through this lipid membrane. I'll illustrate glucose using a blue bead, and then things like amino acids, and the list goes on. Now, if glucose or sodium were to try to get through the membrane, they would find it to be a barrier. Again, selectively permeable. And so they're going to need a passageway. Sodium has a small channel, essentially like a small hole, little tunnel, right through the membrane, whereas glucose needs a carrier protein. Both of these are proteins. This one's usually a bit larger, often depicted somewhat in this way, and that allows them a passage to get through the membrane. So if we had a high concentration of sodium outside the cell, sodium wants to spread out. It wants to diffuse. And if some of it finds a passage into the cell, like through that channel, then it will. And it will continue to diffuse, some in, some away from the cell, but all of it spreading out all the time until the concentration is even inside and outside the cell. So that is facilitated diffusion of sodium. Our channel protein is what facilitated that movement. Glucose, same thing. Let's say we had a bunch of glucose outside the cell. It moves around, spreads out. Some of it gets carried in through this carrier protein. Again, this does not require energy. It simply spreads out in the watery solution. And this can work in both directions, mind you. It doesn't have to be into the cell. Let's say we made a bunch of glucose. Let me borrow a couple from out here. And this cell is rich in glucose inside. Well, the same thing would happen Glucose would spread out. Some of it would find that carrier protein and move out of the cell. This guy over here is trying to get out, but he can't because the membrane is a barrier, so he stays in. When the concentration is even, then the net movement stops, which means that if one comes in, one goes out, there's not really any net movement one direction or the other. So that covers simple diffusion. It covers facilitated diffusion. And then we have osmosis, which is our last type of passive transport. This one simply deals with water moving in and out. It's a bit of a combination of simple and facilitated. We'll take osmosis on in a different lecture.